Welcome everyone. I am very excited to have our first live lecture. Hopefully it is not horrible. I hope you're taking in the lovely view from Angel Island, which is where probably a lot of us wish we could be at. And yeah, if you could just give me a quick hello via the group chat, that would be great to make sure this is actually working and I'm not talking to an endless void. It looks like there are 67 participants there. Still, oh, okay. All right, let's try that again. Welcome everyone, and I hope you're enjoying the amazing view from Angel Island. I think I was talking to no one earlier. I think you guys are in the waiting room, so I'm still definitely.
For some reason, we can't see your face at all. That's interesting.
All right, hang on. 69 participants as of this message. Great. Please chime in and say hi via the chat room if you have a chance. Okay, great. All right, we are live. This is working. Okay. Okay. Oh my gosh, there's so many. All right, so let's try and get started here.
You can partially see your face, but it's kind of obscured by the picture.
Yeah, that is, I was just trying to get a little creative with the setting. Now we're gonna go back to the webcam. So let's see, whoa. Nope, that's not good. Oh, hang on, sorry, I think I know what to do. There we go, great, okay. Hopefully everyone can see me now. Okay, great. Yes. Will Wilson has a very interesting video going on. But sorry.
That's actually okay.
Okay. Great. Yes, everyone can record this. Sorry. Oh, you can't hear me? Some people can't hear me. Yeah. Looks like most people can hear me.
It says we need recording permission from the host.
Okay. Let me look for that. And yeah, thank you everyone for bearing with me. This is my first live, well, my first virtual live lecture ever. Let's see. Reactions. Does anyone know where record permissions might be? It's automatically recorded. Okay, yes. It's automatically being recorded. I have that setting going. So I'm not gonna try to figure that out right now. But I will post this as well. So for all those who can't make it, although 73 out of 109 is pretty good. So I will post this afterwards so you can watch it as many times as you want. Okay, yes. So, oh my gosh.
So will you post it as a YouTube video link or a file?
Probably both, but definitely whatever is fastest. I haven't done this from Zoom before, but I think I can just do it as YouTube. So I honestly don't know for sure, but I think YouTube is the best idea because then it will automatically caption. So that's a more accessible document for everyone. And yeah, I'm just gonna, let's just take a couple minutes for other general questions or specific ones. I just wanna make sure that we're all good to go before we start. Okay, micro minutes, I see. Yeah, so for the micro minutes, sorry if that wasn't clear. Oh, I need to admit more people. Jeez. Okay. Whoops. All right. So the micro minutes is just a discussion. So there's sort of like I'm transitioning these assignments, at least for right now, because it's a paper reading on answering the question. So basically, I'm posting in questions to prepare everyone for the exam format. So basically, we have an exam on Thursday of next week, and instead of just giving you microminute questions, I'm trying to like adapt the questions that we do that will be closer to the exam, because I think that will give people more practice and they'll feel more comfortable in completing the exam. So it's not the like standard answer these 3 questions that are multiple choice and then see what happens. Okay, other questions. I can set it so it automatically admits people. That would be great. Thank you. I don't know where that setting is, but yeah, let's see more. Allow participants.
Also, how will our project work? That one where we get to choose our own topic?
Yeah, hang on. There's a lot of questions in the chat too, Carlos.
Oh, okay.
But 1 second.
Pardon me.
It's okay. Zoom has captioning service as well. Okay, good to know. Zoom has captioning. Okay, I'll look into that. Is there microminutes in online or it's just discussions on Canvas? Okay, that's to Armin. Can you set? You can set it. I don't know how to set it. I'll look into that. Auto set admit. This is all very helpful, by the way, and I'm sorry I don't know all this. I've taken a lot of webinars over the last week, but they've been on a wide range of things. Is the 317 assignment extended to tomorrow? Yes, it should be. So the assignment that I posted yesterday on the questions that are supposed to be posted on the discussion board, those are supposed to be due Friday at 5 p.m. I'm pretty sure that's what I said to that, but I'll check that as well. Okay, and then we have, yeah, so that's extended. So is there going to be a post-lecture or assignment for this class? Yes, there will be an additional lecture assignment for this class. And again, it's just to get everyone more familiar with the material and how that would like appear in an exam format, especially because this is our first primary paper. So yes, there will be a post 319 assignment. Okay. Are we still going to be doing the micromolds project? Wow, people don't want to do the micromolds project? Yes, we're definitely still going to do the micromolds project, except there may be a couple of changes. But what'll be great is that when I've done similar versions of this project before, everyone recorded a 1-minute video. And so really, like, online is actually very conducive to 1-minute videos. So that's, I think, one of the strengths of the project. And we'll discuss that as well, but I'll probably wait till after. We'll probably do that the first day after spring break, or first class after spring break, just because I really want to make sure everyone feels good and comfortable with the exam, especially since we've transitioned to this online format. I don't know where it is. I only know the other professor I have it set that way. Okay. Yes, that's okay. Great. And then more messages. It is still 5 p.m. Thank you, Demi. Great. Will we get the keys? Oh, the keys. Yes, after everyone's submitted, I will post the answer key for sure. Answer key for sure. I have to say, I definitely missed everyone, and actually getting to see your names in real time chatting brings a bit of the community aspect back, so that's good. Where do we find the post-lecture assignments? Those are all posted in the modules. So for every class module now, oh my gosh. All right, yes, I'm glad some of you missed me, that is great. What I was trying to say is that the modules are going to all contain whatever post-assignment lectures, the class notes, and the discussion boards for like the photo 19, etc. Okay, so that's basically what I'm going to try and structure things as. And if there's confusion, please just email me. I try to still get back to everyone's emails in 24 hours. And then I also, sometimes if I don't respond right away, it's because it's going to go out to an update.
Yeah, that was my question actually. So you consolidated everything, basically the discussion boards, the post-lecture assignments, everything is going to be on the module?
Yes, yes, it's all going to be on the modules now.
Okay.
We can virtually laugh for your jokes now. Yes, that is true. Okay, great. Thank you. This is really good feedback real time, because when I was doing the pre-recorded lecture, I really didn't have this, so that's good. Okay, I had a couple general announcements. I'm sure there's a way to do this, but let's see. Breakout rooms, share screen, video. I don't remember if there's I forget if it was this or a different app to do a like quick poll, but reactions, no. All right, maybe not. Manage speaker view. Okay, well, share screen. Share screen, show all windows, advanced. Portion of screen, content, files. I do think you have the option to do connect full by node, yeah. Okay, I thought it might be easy, but yeah, I guess not. Okay, so anyway, I'll post a couple polls after this, just again for me to get more feedback on things, but sorry, hang on. Okay, so, ooh, okay. What's going on? Let's try this. Nope, sorry. Okay, can everyone see class slides 319, hopefully? I don't know, don't you wait, oh, damn. Okay, sorry for that. Right, okay. Yes, lots of yeses, great, okay. It's actually scary dealing with this because it's like a force of nature, like it's like 100 people and then you get 20 yeses. Okay, so after the lecture, or after whatever we want to call this, virtual online in my living room, I'll ask, just think about if you like this version better or the pre-recorded version better. The other question I'll have is if you thought Tuesday's lecture was too long. I did not intend for it to be that long, but that's what it was. So yeah, and then I might have a couple other questions too. So just look out for the polls. I really appreciate your feedback. And then today's lecture is going to be a little shorter. well, we'll see how it goes, but I tried to keep it light because I felt bad about how long on Tuesdays went, and I don't want to give you a bunch of information in this new format before an exam or a new format. And then basically my thought was for Tuesday's class, we could just try doing like a mini exam on sort of this entire unit, which I feel like is very disjointed because of the COVID-19 stuff. And I just want everyone to feel sort of prepared. And so trying to pull questions from like the lac operon or DNA replication, everyone's favorite topic. Yeah, like a review session. Do you mean a micro? Nice. See, you guys need to be this vocal in class. Like that was an amazing, a micro exam. Yeah, so I actually, when I taught this class before, called it a mini exam, and then I had a picture of Minnie Mouse in an exam, but that's just like random. Micro exam makes a lot more sense. Okay. All right, I think that's all for general announcements. So let's get started. The class slides are online. Sorry, I posted them earlier, but then I forgot to publish them, which to you guys looks like the same. So yeah, I will, yeah, try to be better about that. Okay, so let's get started. Okay, so I wanted to touch base on the review question from last time, or I guess the open thought question. I showed everyone the answer at the very end, and hopefully you had a chance to work through this. Let's just, because I don't really know how to do these polls, can we think about, let's see. Yeah, could you just say like, just say yes or no if you did this question, just so I have a sense. Oh, okay, yeah, I'm definitely gonna have to look into a poll situation. Okay, no, not yet. Okay, well, I'm glad at least some of you have done it, but basically I'm gonna walk through the logic behind this because the idea is that this will be similar to things that you'll see on the exam. So basically you're measuring the expression of different host receptors on the Vero cells and get the results to the right. And sorry, I can't see the results to the right. Okay, great. So you're looking at just this expression of different host receptors. And all of these host receptors were discussed to some capacity in the paper. And then next you decide to infect the primary Vero cells that are pre-incubated with 0 to 20 or 40 micrograms of this anti-ACE2 antibody. Oh wow, typo, sorry. The cells are the Shakespearean version, are inoculated with pseudotyped VSV harboring this human coronavirus 299ES. So that's spike protein, okay? And then at 16 hours, the pseudotype entry was analyzed. What are your expected results, and explain your rationale? So what we have here is basically an experiment that is similar to the one that is shown in the paper, and really is pulling from both figure 3B with regard to the antibody use, and then also figure 3A with the other coronavirus. And for the actual answer, you would not expect to see pseudotype entry. And this is because you want to think about what is the requirement for pseudotype entry for the 299E spike protein in that coronavirus. And if you remember from the reading in the paper, it talks about how the entry receptor is actually the APN receptor. which is not expressed really by Bureau cells based on the data that we see in the top right corner. So you would not expect any entry or very low level of entry. So that's one approach for trying to navigate these kinds of questions. And I encourage everyone to think about the requirements for pseudotype entry, as well as what pseudotype entry actually is, which we're going to go into a lot of depth on today. Originally, I was thinking we would just spend time covering all the figures in detail, but I have been talking to people both via email and office hours, and I think there's actually some confusion on that, and it's a really important concept that I want to make sure we're all on the same page on. And it can be quite confusing too. But since most of the paper uses pseudotype entry, I really want to make sure we address that. Okay, so I'm sorry. I'm just trying to see if I can like rearrange the view a little bit here. Okay, I'm just gonna do this. All right, great, thanks. Okay, so, and so I can just include these figures again. I know I went over them at the tail end of the last lecture, but this is sort of the figures that you'd want to focus on in terms of tackling that first question. Oh, yes, great. Sherry, yes, it is similar to figure 3 and B. So I'm glad we have that on the same wavelength there. And then figure 3C wasn't really part of that question, but you could also think of ways where you'd want to apply these findings, and really, as I mentioned last time, this is the authentic version of the virus, which really is sort of a proof of concept thing. And you again see a consistent phenotype that's dependent on ACE2. So now we're going to move on to figure 4, okay? And so there's multiple panels here, and you are now you're still looking at pseudotype entry, but now you're looking at it either in the presence or the absence of the TMPRSS2 host protein, which we should remember has a priming function, so it modifies the spike 2 formation. Sorry, waiting room. Okay. And so that is important for the entry phenotype. Once again, we're looking at 3 pseudotypes. So we have VSVG, SARS-S, and SARS-2S, okay? And then we're looking at a couple other experimental conditions. Who can tell me what the function of E64D is, or camostat, or the combination? What is the point of adding those? And you can chat. You can chat. Which one blocks ACE? Okay, great. And then what is E64D doing? You can check the paper if you want. Come on, there are 79 people. What is E64D? Okay, good. So it's an inhibitor of cathepsin B and L, all right? And these are endosomal cysteine proteases. So, okay, great. Yeah, okay, people are getting it now. So we have these two different inhibitors that they're using, and then we're looking at pseudotype entry. So based on the graph, so if you look at page 3 of the primary paper, basically on the second column, it describes how you have SARS coronavirus 2 having the capacity to use the endosomal cysteine proteases. And then it goes on to describe different things that can block that. So basically that whole column is where the information is in the paper. So based on this figure, what do you think one of the main points from panel A is? Do we think, which condition do we think is more effective at limiting, or which condition do we think is most effective at limiting pseudotype entry?
So would it be the combination of E64D and chemostat?
Sorry, I'm reading a lot here. Yeah, so if you look at panel A in the top portion, you see that there is pseudotype entry that occurs for both SARS-S and SARS-2S when you have the chemostat inhibitor. But in the bottom, you see that it's actually most effective when you have the combination. And it seems like both, or a general amount of the class sees that trend as well. So basically, the higher the bar, the more pseudotype entry you're getting. You see that both for the SARS S and SARS-2S pseudotypes, you get the most robust inhibition of this when you add both of the inhibitors. And so that suggests that there is, again, sort of a high conservation from a biological standpoint of what these inhibitors, of what these two coronaviruses are using to cause infection. So let's see, let's move on to the next panel then. Here, they're again looking at the same three pseudotypes. And now they're modifying different variables, either TMPRSS2 or E64D. And okay, sorry, question. So basically, you want to think about the mechanism of action and then how that applies to the graph that you're looking at. So if you're thinking about cysteine proteases, those are cathepsin B and L. And then you want to differentiate that between the serine protease, which is TMR, PR, SS2. If you are able to block whatever you're able to block with those inhibitors, is then that determines the specificity, if that makes sense. Okay, so we'll circle back to that, but first let's get through this panel. So what do we think is the main point about pseudotype entry with regard to this? Is TMPRSS2 important? Is that presence important for successful pseudotype entry? Highly crucial, yes. Is it more important for one coronavirus versus the other? And what about VSG or VSVG? Okay, good. So it looks like people are getting these concepts, which is good. It was our first primary paper together, so I wasn't sure exactly how tricky this is going to be. Okay, so now they're doing titrations, okay, of the camostat inhibitor, and you have no inhibition of VSVG. Again, it's not a coronavirus, so you might not expect that. Its mechanism of action is different. However, if you look at the 3 coronaviruses, you have this very clear dose-dependent inhibition over a range of concentrations, and basically it trends to undetectable levels after about 5 to 10 micromolars, depending on the coronavirus. And so this is just, again, clear. Yeah, that's a good question. I guess we didn't really talk about positive and negative controls. I apologize for that. In every experiment, you should design a positive and negative control, ideally. If you, if you look at this graph, the VSVG panel would be the positive control. Because its mechanism of action is different, you see clear pseudotype entry regardless of the amount of camostat that you add. Okay, so okay. All right, so yes, if we ever return to real class, although I guess we probably are not, you should definitely talk like this, like in the classroom, because it's hilarious. But basically, what would be the negative? What do we think the negative control is? Okay, so actually, yeah. So I'll take a step back because okay, everyone needs to calm down. All right, so basically, this graph is a little confusing, and I think I actually misspoke earlier. So basically, when you design an experiment, you want to think of something that would give you it. When you're designing an experiment, you have your control variables and then your experimental variables. And when you think of your negative controls, that means that if nothing happens to the system, if nothing is happening, then that is your negative. So most commonly, this is when you add buffer to a system, and then in the experimental condition, you're adding an inhibitor or a virus or bacteria. So all of these titrations, excuse me, have an internal negative control, which is the 0 condition, that gray bar, okay? So if you look, the gray bar at every instance is the highest. The positive control doesn't always mean it's the highest one. It means it's the one that has no agent of change present. So there's, in fact, negative controls for the VSVG titration, right? So that would serve as a negative control. And same with MERS, same with SARS, same with SARS-2. So all of those gray bars serve as independent negative controls for each of the viral subgroups. Then, depending on how you frame it, depending on the question you're asking, that dictates what the positive control is. So the positive control can be tricky to define, but basically it is a known result that has already been validated in a system. So oftentimes that's in a previously published paper or previously known knowledge, right? So in this instance, you can make the case that SARS-S is a good positive control because it's already known that camostat is an inhibitor for it. And so because it is already known that SARS-S is an inhibitor for it, you know what result to expect, okay? You know what you're going to see, or you would expect to see, as long as the experiment is performed correctly, which is this dose-dependent inhibition. The experimental one here is really SARS-2S. It's what the paper is centered on, and this pseudotype has not yet been tested. But because you have now a good positive and negative control, you can compare and see, is it closer to the negative control or is it closer to the positive control? And thus, you're able to have your experimental variables and compare them to your controls because it gives you a frame of reference. Because you've established this frame of reference between your positive and negative controls, you see that it pretty clearly reflects the positive control, which is SARS-S, and so you can make a determination as to how profound this effect is. So hopefully that makes sense, and if not, again, I'm having office hours after this and we can talk about that further. So, okay, in the interest of time, we're gonna move on to this next panel. Okay, so what we have here is figure 4D, and you have the real authentic SARS coronavirus 2 being used here in the presence or absence of an inhibitor. And so you are able to, again, see a clear phenotype, this time at 24 hours when you have the inhibitor present. Okay, so they're looking at genomic equivalence, and they measure this by qRT-PCR. And this is a different axes. This is a different measurement than the previous graphs because the system is slightly different. They're no longer using pseudotypes, so they actually have to measure by priming up the genomic material that is present from the virus and then using that as a readout. In the previous ones, they use a Firefly luciferase reporter, which they're able to read with a luminescent plate reader. There are also other readouts that you can use. So again, this is consistent with their model that the camostat inhibitor plays an important role. Whoa. Whoops, sorry, I clicked the wrong thing here. Oh, that's very nice. Okay. All right, and then now they're doing pseudotype entry again with the chemostat inhibitor at different concentrations compared to the SVG. Does anyone want to comment on what the main point of this figure is? Did I put you guys to sleep? Okay, good. Okay, great. Well, yeah, basically, again, looking at chemostat inhibition of entry, both with SARS and SARS-2S, they're both significant. You can make a case that the error bars are slightly larger with the SARS-2S, but they also have a relatively small sample size here. And I just want to double check something. Right. So why did they do this experiment if they already showed us chemostat was important for pseudotype entry? What? Why? Experiments cost money, they don't get you money. We just went through panels looking at how awesome chemostat is inhibiting pseudotype entry. Effective as a treatment. VSVG is the envelope protein for the vesicular stomatitis virus. Effective as a treatment, okay, but again, what isn't panel C enough? I mean, they show this pretty extensively with chemostat here. Why would they just do it again? Mechanism of action? I mean, it's chemostat, it's the virus, it's chemostat, it's the virus. Well, it looks like they do, is it to find the right dosage? I mean, look at all the doses they did here. If anything, they got lazy here, right? Right, but like, so what else could be different? They have the same viruses. They are doing it in a pseudotype form. They're doing the same concentrations of chemostat, if you look at 10 and 50. And they're doing this with just a smaller sample population. All right, well, you guys have given good guesses, so not sure what you mean by basic amino acids. So there are differences between SARS-S and SARS-2S, the spike protein, but again, that's all used right here. So the same spike protein is being used in C and E. If you look carefully, let me find this really quick. Sorry, I thought I had it. 4D. Right, so, in C, they're using KLU3 cells, okay? But KLU3 cells, oh, sorry, you guys are chatting a lot. which is good. Conversation is good. But the difference that I was trying to highlight is the host side of things. Right now, we're really focused to see protease activity, right? So yeah, but really the host side here, okay, has changed. Before, they're using a cell line that's been incubated with these different concentrations, but the cell line is not a human, and it's not reflecting the host interface that these viruses are going to encounter. Specifically, what they change here in the figure 3 is that they're looking at human lung cells, specifically primary human airway epithelial cells, because this is more reflective of the human host. So it's great to demonstrate things in lab. We need to start somewhere, but ultimately, if we want to know if these reagents or inhibitors are going to be effective in humans, we need to start to move closer to a human system. And primary lung epithelial cells, yes, E is more reflective of the human host. So yes, and so and Tommy's bringing up the point that they use a whole range of cell lines. So they use human epithelial kidney cells, they use canine cells, they use like all sorts of cell lines. But ultimately, humans care the most about curing humans, unless you like really love your dog more than you. And I mean, which is fine, but you know, then your dog might survive and you won't. But basically, by and large, human research is set to cure humans. That's the goal. That's why the funding is put in place. We do a lot of intermediate steps to get there, but moving closer to it is looking at a primary human cell line. So these cells were taken from lung biopsies that would then allow you to have a more accurate readout for what would happen if the virus were to infect humans, which is what it's doing, right? So that makes their argument stronger. If this doesn't work in primary human lung cells, then you sort of lose efficacy in that context. Okay, so let's see here. Okay. Right, and then we're going to move on to figure 5, part 1. And you see that here they're showing inhibition of pseudotype entry again, but this time they're doing it with patient serum from convalescent patients, okay? So they previously had SARS, the original, and then they're able to have some efficacy against the pseudotype entry. You'll notice that the SARS S pseudotype entry is in general more dramatically inhibited compared to the SARS-2S entry. So only at these really high dilutions do you start to see an effect in terms of providing protection. If we look at the second part, the trend is pretty consistent. And we also see something similar when they used rabbit serum. So this is sort of, again, another step that's closer. Now they're using patient serum, and this could be able to guide therapeutic efforts as we try to figure out how we can limit the spread and infection of humans across the globe. So this is probably one of the biggest reasons they got into Cell, which is a really good journal. They're able to demonstrate some effect using patient human serum. And while it is not as protective, it again brings us that much closer to understanding mechanism of action and seeing it with a human component that we could use in the clinic. Things that they did not show are doing this with the real virus. And that could be because maybe they just wanted to publish what they had, or maybe they didn't see significant results, or maybe they couldn't get the assay to work in time. There's a number of reasons, but we don't really know for sure. So you always want to think about what they're showing and what they're not showing you, because going back to differences between textbooks and primary papers, this is something that, this is a more fluid document, and it doesn't mean it's perfect, right? Like they might be cutting edge, but there could be gaps or things that don't make sense. And sometimes that's because of experimental constraints, and sometimes it's because they couldn't get things to work. So that is always something you want to think about in terms of the context of the information. Okay, so now we're going to switch gears a little bit and talk about one of the methods, which is pseudotyping. This is used extensively throughout the paper. If you look at the y-axis, most of the time it's looking at pseudotype entry. What is a reason they might use pseudotype entry instead of the real virus? We talked about why they might use a real virus instead of pseudotype entry, but what about the reverse? And you can just feel free to chat out your answers. Hello. What was the question? Yes, okay, whoa. Okay, so hang on, what was the question? Isolate specific protein, can you type out the question? Oh, that's a good idea, thank you. Yes, if there are things they can be doing better, I am all ears for that. Why use, oh, all right, yep, okay, yes, why use pseudotype entry instead of real virus? Real viruses are more, pseudotypes are more understood and safer. Yes, great. So safety is an important aspect, right? The reason that coronaviruses are a big deal is because it's spreading all over the world and very dangerous. Less dangerous to work with, right? There is another, yes, scary, could be a Halloween costume. So basically, you're able to look at a specific protein, study it in a different system that is not as dangerous. The VSV virus is classified as BSL-1. So that's like the lowest biosafety level that you can have. Other considerations are that not all viruses have a high titer load. So if you want to grow up a lot of the virus, sometimes viruses such as rabies and some of the hepatitis viruses have a very low titer production. So it's very tricky to generate a lot of the virus, which then makes it hard to do your infection experiments. So from an experimental standpoint, there are advantages using VSV, which produces a lot of viral titer. And so you can use a lot of the pseudotype virus to then perform your experiments. Can we put that question on the exam? Maybe, why not? I'm not saying yes, I'm not saying no, but yes, I mean, those are similar kinds of questions, and hopefully you had a chance to practice those in the pre-recorded lecture as well, but there's gonna be some graphing questions, but then there's just gonna be some like thought questions and reflection questions, like what does this graph show you? Or why did they choose this instead of that? So like, no one responded. Well, people responded, people guessed as to why they did 4E, but you know, there again, that's a good example of a question. It was like, well, they're not just repeating it because they're bored, they're repeating it because they're changing a variable. What is that variable? Okay, I did not say it was gonna be on the exam, but okay, so don't worry. And this is all recorded, so you can literally just watch this 1,000 times. Okay, so, right, so we've covered reasons for why they would pseudotype. Whoa, okay. I need to put a clock in this room. Okay, but here we have pseudotyping, okay? So basically, this is the VSV virus, or this is VSV, yeah, last part of this virus, okay? This is what it looks like. It has a lipid bilayer, it has glycoproteins, that's the G protein, okay, that's the key envelope protein. It has an LMP protein, that's RNA polymerase. It also has matrix proteins. You see this negative stranded RNA genome, and then nucleocapsids, which protect the genome, okay? So it basically looks bullet shaped, and I made a cartoon image on the right here. And now we're gonna go over all of, well, not all of, but the basic idea of pseudotyping. So if you have this wild type virus, which of the proteins below would you expect it to have? So I have two columns, it's the haves and the have-nots. What are we looking at again? What does VSV mean? What? Okay, so I encourage everyone to read the paper because this is key. This is a key aspect of the paper. But basically, this is the virus that they use for pseudotyping, okay? And let me see if I can find the exact place it says it. Maybe it doesn't say it. Well, okay, if you don't know what something is, please always look it up, okay? But it stands for vesicular stomatitis virus. I'm not sure if I'm saying it exactly correctly. I thought I could find it exactly in the paper, but vesicular stomatitis virus. Okay, so write this down on the paper, and that will be a good note. But like that's what you should do for every term you don't know. Okay. Oh, good job. You guys are answering that. Okay, so VSV has all the VSV proteins and does not have Firefly or EGFP. Great. So that's correct. Basically, you want to see this as a original unaltered virus, and so Firefly and GFP are things that we added in to manipulate it later in the paper. If you look at this infection, you have the wild type infection, and then deposits the negative strand RNA. You then have viral protein production, and then the envelope forms and you have envelope formation, and then budding occurs. So this is like a basic standard viral life cycle. Now we want to know what happens if you inoculate with the VSV delta G F luciferase infection, okay? So I want you to think about what this has and what this does not have, both in terms of RNA and protein. And this is my attempt at, okay, so first I want you to draw what this looks like. And you're gonna do this in groups. So, let's see here. Groups, breakout rooms. Okay, great. So 83 participants into, wow, this is cool. Okay. Like Dr. Adams, you're easily amused. Okay, here we go. So, creating rooms, open all rooms, I guess. Whoa, this is really cool. Okay, we're gonna open all the rooms. Okay, so join your breakout rooms and then also look at the instructions. Okay, Okay, the instructions, sorry. So first, go to this link. It's sketch.iopad or .io.sketchpad, okay. And then draw what this looks like, okay. And you can use the wild type image as a reference, okay. And then you can type out in the sketchpad, there's a type function, what your image looks like. Has entered the waiting room, okay. And then I want you to post your screenshots in your group breakout sessions. And then we're gonna post these on a discussion board in Canvas, but we can do that after class. But basically, I want you to post your screenshots to your group so you can have a group discussion about what you think it looks like. And I'm gonna also try to demonstrate this, but, whoa. Oh, and I can join.
Dr. Adams.
Oh, hello, how's it going?
Hey, so what, we don't know, you said referencing instructions, but we don't know what to do.
So can you see the instructions on like 1 through 5 on the main screen?
Oh,
5 on the screen.
you guys can see
No.
the main screen?
No, we just see our breakout room.
Let's see. Let me try this. Okay, sorry, I've not used breakout rooms before. Can you see the instructions now?
Yes,
Okay,
we can see it.
great. So you want to go to this website and sketch it out, and then think about what RNA and protein are also present.
Sketch.io. That sounds like a game website.
People
Like
might use it for
snake.io.
games. Okay, so take a screenshot of the instructions if you can. I'm going to jump to another breakout room and see if I can help them. Okay.
I got it.
He wants us to solve this one around. Oh,
Hey.
never mind.
Walter!
Hello, Demi. How are you?
Good, how are you?
Good. Good to see you.
Me too. We are having hectic. We forgot what you asked.
It's okay. So if you can see my screen, you can now see, you should now be able to see the instructions.
Okay.
Oh,
Yeah.
So go there and
Okay.
then start the drawing process.
Sketch. Okay, let's see what I could do.
Okay. Have you taken a screenshot of this for your group?
wait.
Oh, that's a good. No, I didn't screenshot.
Okay, I took a picture.
Okay, cool. Thank you.
Okay, you're welcome. All right. Let me know if you need something.
Okay. Okay.
All right. Okay.
How long do we have? Did you limit us to a time?
Try to do this in 5 minutes so then we can share out.
Okay, so we'll leave the break room in 5. Okay. Okay, guys. I'm gonna go to the website. Sketch.io.sketchpad. Oh, I should probably share this with you guys. Crap. How do you do that?
Does only one of us need to do the sketch thing?
Yeah.
Do all of
I mean, if you, I want everyone to be, I encourage everyone to try it, but if you're, don't want to do it, that's totally fine. I thought this is a fun interactive thing that we normally wouldn't be able to do in class. So yeah, I'll leave it up to you, but definitely try to figure out what you think they have and what you don't think they have.
Okay, we haven't
And this is
heard
just an
any-
easy way to get participation on the discussion board as well.
Okay, thank you.
Okay. All right, do we have any feedback from Juan or Michael or Emily? You guys haven't said anything at all.
Oh, I am Emily. I am here.
Oh, cool.
I don't know
Okay,
if you
what about
can hear me
Michael?
or not.
Yeah, I can hear you guys now. There's just a couple of you guys are on mute still, so it's kind of hard to work with a group if you guys are on mute, you know.
Michael
I'm here.
Oyang.
I'm here.
Okay.
Oh, no, you're
Wait, he's not even here.
Wait, William, what's wrong? Wait, can you explain your webcam? I don't get it.
So my computer's webcam is broken. So instead, it's just streaming initial D. And the computer thinks that that's in the webcam, and that's what it's recording.
That's so funny.
So now it's just forever going to stream.
So what if you turn off your webcam?
What?
What if you turn off your webcam?
I guess it would just show a black screen.
Try it.
Actually, I have no idea how to turn that off.
It's on the bottom, right? Are you on your laptop?
The bottom.
Where to stop video, or start video?
Oh, if you click stop video, it's just going to show nothing at all, because that's what do you think stop video means?
Yeah, no, I'm just wondering if your webcam is just still gonna stream it, you know?
It shouldn't.
If you press on stop video, it doesn't stop the meeting.
Well, if it's, oh, I see what you mean. Okay.
Yeah.
Yeah, now
it
we
just
see it
stops.
streaming. Yeah, exactly.
I figured.
Yeah, so we don't have to see this streaming, you know?
Yo, does anyone know what we were supposed to draw and what it was supposed to look like?
I'm trying my best, but my drawing skills are not very well.
Did you post it? What were we supposed to post it to?
I think it's in the lecture.
Your mic is muted.
It does not have to be a masterpiece, thank you. Yes, I just was checking to make sure the discussion board is up for the posting, since we are unfortunately running out of time, although I'm fine going a couple minutes over since we had a slow start, but I also realize everyone has a schedule to stick with. So hopefully you had at least an opportunity to try and sketch something. You can also continue this after, but for now, I'm gonna go back to, let's see, I'm gonna close all rooms and we're gonna go to the actual ideas behind this. So I guess everyone has 60 seconds to leave your rooms. Okay. So everyone should leave their rooms, and then I'm gonna walk us through a couple of the different mechanisms here. So basically, one idea could be that maybe you drew a VSV delta G F luciferase virus. Oh, why is it not a? Oh crap. Okay, sorry. I think I'm in the class slides. Yep, that would be it. Okay, 1 second.
Hey Walter, were we supposed to upload them somewhere? And to be honest, we were having a really hard time interpreting. So I did my best to draw with a feather pen.
Okay, it's okay.
So
I will probably just
Good.
add this to our post-class assessment. So
Okay.
don't stress if you did not get a perfect thing. Okay. So basically, one option is that you could draw the VSV delta G F luciferase pseudotype with having all of this RNA, all of these RNA components present, and lacking the delta G virus, delta G protein. And then you could say it doesn't have any of these other proteins, but it does have these. And if you...
Where are you pointing at?
Oh, can you not see my arrow?
No, I'm looking at your face.
Oh, sorry.
The slides.
Sorry, I forgot to share the screen. Okay. 1 second. Okay.
And can you kind of walk us through step by step from the beginning?
Yeah, sorry, I just thought my screen was shared, so I apologize. Okay, so okay, so if you have to go, I totally understand, but I'm going to keep going because this is recorded and I want you to have this information. But basically, one idea is that, okay, sorry. Okay, can people see the screen now?
Yes.
Okay, great. So one option is that that you have selected all of these proteins to be present. So you have 3 proteins that are selected and then 5 of the RNAs that are selected, okay? And basically you would have a pseudotype virus that looks like this. So it lacks that G outer protein, which is the envelope protein. And then if you carry this virus forward, you see that there is, in fact, no way for the virus to enter the host cells because the envelope protein is what mediates the attachment. So
what does delta G mean again?
That means that it lacks the G protein, the envelope protein. So if we go back to the previous slide, our wild type protein has these G proteins, which are my cartoon representation of the glycoproteins that are surrounding the VSV image. So now if we go forward and we're looking at this possible idea, if it lacks the VSVG, then it's not able to enter the cell because it can't use its membrane with it. So this idea would not work for generating pseudotypes because there is not that fusion present. It goes off into the abyss, and we're ruling this out. We could think of other manipulations here where maybe the VSV has different components. So a second idea could be that instead we say that it now contains the VSV-G RNA. Okay, so it has the RNA to make it, but it still lacks the protein. Again, this idea would not work because you're lacking the glycoprotein, which is required for this fusion to occur. So you're unable to get entry into the host cell. With no entry possible, again, your virus will fade into the abyss or this pseudotype, and this option will not work. So then we could propose yet another option, okay? This idea is that it contains the VSV-G protein, but it lacks the RNA to encode the protein. So here we have the spikes, okay? We have the glycoprotein that is present around the membrane, but it lacks the RNA to encode it. Here we have the binding and fusion, as well as the release of the viral RNA into the host cell. This means that it's then able to proceed, and you have production of viral proteins, but importantly, you do not have production of the glycoprotein, right? And that is because it's not encoded. That is the mutant component. So once we have production of these viral proteins, are there any guesses as to what we think might happen?
They can replicate to infect new cells.
Do we agree with that or do we disagree with that? Okay, sorry, I'm having trouble viewing the chat room, which is really annoying. Okay, well, so you could think that yes, you're going to have formation of the envelope and budding occur. However, we have to remember one very important thing, which is that this virus lacks something. What does the virus lack? It lacks, oh, now I have the chat back. Okay, it lacks the glycoprotein, right? So can this virus form without the glycoprotein?
It can, but
no, it cannot. It needs the glycoprotein. It's really important for the envelope structure. So
I thought that was a membrane protein that was.
Yeah, so it can vary depending on the virus, but for VSV, they need the glycoproteins.
Right, because to my understanding, the previous slide you showed that there's viruses that doesn't have the glycoprotein and it's still forming. So that makes me think that it can still form, but it's formed without the glycoprotein around it, if you know what I'm saying.
That's a very good point. Yes, so in that illustration, that's actually very misleading. So I apologize for that. Technically, that virus is not viable. So it's really, the virus would degrade if that was the only structure it had. I was trying to impress upon the class that if that's what you drew, I was trying to walk through the logic of that model. But yes, you're bringing up a very good point. Not only would it not be able to bind to the host cells, it would really just cease to exist because it lacks this key structural component for its membrane. So thank you for raising that. With regard to proceeding with this, because it lacks the ability to encode the glycoprotein, it is unable to generate these. So in fact, this is not possible, and you would not have budding. So once again, you would not have the ability to generate the pseudotype. So we have collectively tried to generate virus without the glycoprotein, generate it without the RNA, generate it without both of them. So all of these methods are not working. Is there an idea that people have which would allow us to generate the pseudotype? Is there some additional modification or manipulation of this experimental system that allow us to generate it? I tricked you. Okay. It's a thought experiment. Right, so if it has both the protein and the RNA for the VSVG, then what's the difference between it and wild type? We need both. We need both. For wild type has both, so this has both. So if it lacks the other VSV proteins, are you able to analyze the requirement for these glycobinding or these glycoproteins that mediate binding? Because isn't that what we're interested in?
Wait, can you repeat the question again?
Sure. So what manipulation could you make to this experimental system? I'm saying it slowly because I'm typing it out at the same time. What manipulation could you make to this experimental system to allow for the production of viral, of pseudotype virus, specifically this VSV asterisk delta G. Okay. So I really personally miss this. So for everyone who wants to stick with it, like I'm gonna do another breakout room, and if people, just to give you all a chance to discuss this, because I think this is an important concept. And we're just gonna do this for like a couple minutes and then we'll be wrapping up, but we're gonna do this one more time. Let's see if I can. Breakout rooms close automatically, and oh, for less time though? Yes, definitely. Okay, well, let's do 2 minutes to discuss what manipulation could you add. And the hint is like, consider modifying the virus and another parameter that is not the virus. Okay, and here we go.
Virus, and I think for the thing that's not the virus, you can change the cell itself that it's infecting, like the receptors.
How is this group doing?
I was thinking
We're kind of reviewing what we were talking about.
Yeah.
Yeah.
You said change something that's not the virus, right? So possibly the host cell's receptors?
Right. So that's a good idea. You can modify the host environment possibly also by adding something to it, right? So in the paper, they talk a lot about adding in the protein of interest. So what if you were to supply the virus with that protein of interest, right? So if you were to have it present in the host cell, then that might be a way to get around it. And now I think the rooms are closing. Okay, but I'm going to demonstrate this in the main session, and hopefully it'll make a little more sense. I apologize for this being a little more rushed than I wanted, but yes, I hope I will get better at future sessions.
For sure.
Walter, before you proceed, I can't hear you. I think you muted yourself. I can't hear anything said.
Right. Okay. Well, first, it was an apology to everyone that it was so such a mess today. But thank you for bearing with me, and I really, if you give me more chances, I will promise I'll try my best every time. So hope to get better with this. Okay, so that was my apology, and then second was the explanation. So basically, for what the way to navigate this tricky question is how they use it in the paper. So basically, what they did was they were manipulating the host cells by providing them with the viral protein of interest. Okay, so basically what we can see on the shared screen is that they grew up 293Ts, okay? And because they grew up 293Ts alone before the infection, they're then able to transfect them with the viral glycoproteins. So by adding in the missing viral component ahead of time, the cells are now all set to have this additional resource for the virus, okay? So if we now take these cells that have been transfected with a specific viral glycoprotein, and then you inoculate them with the VSV delta G firefly luciferase, you're going to get this binding interaction where the negative strand RNA is inserted into the host cells, and then that genetic material will be produced, okay? So all the viral proteins except the G glycoprotein will be produced by the virus replication machinery. And then the host will be producing the missing glycoprotein with its replication machinery. So this can exist as an extrachromosomal plasmid inside the host cells, and the host machinery will produce this. That protein, the glycoprotein, can then be used for construction of new viral capsids, okay? So the protein is produced by the host does not give the virus the genetic material for the glycoprotein, but it does produce the protein itself. So it's like, it's giving you the instructions, it's giving you like the walls for a house, but it's not giving you the instructions on how to build the house. But the virus doesn't really care. It's just like, I want a house. And so then it now has the house provided to it by the cell. And then the rest of the genetic material that the virus has goes in there. But it still doesn't have the instructions for the glycoprotein. It still doesn't have the ability to create glycoprotein on its own. And that is functionally what a pseudotype is, right? There's a reason that the authors of this paper only look at pseudotype entry, right? Because that's what a pseudotype can do. It can enter into host cells, right? Unless you help it out. It can enter, and that's great. And you can look at like the fluorescence that is generated if it has the firefly luciferase. But if it lacks the ability to create its own capsid with this glycoprotein, then it can't go on to infect things, which is great because that means it's really safe to work with. It's a one-shot wonder. It's one and done. So you can look at entry. You don't have to worry about getting infected. It's just a pseudotype, and it can't go on and replicate indefinitely. So unless you help it out, yeah. Mm-hmm.
Is it is the host cell basically getting infected twice? Like the first time with the glycoproteins, and then the second time with the VSV?
So the first time is a transfection. It's not an infection. So it depends how you do it, but a common way is to use just different reagents, transfection reagents that can deliver the genetic material of a virus to a host cell, but you encode it in a plasmid. So then the host cell recognizes it as its own, and then produces the product, right? It doesn't know that it's going to be producing this viral protein. So you're tricking the host cell into making the viral protein, then the virus comes along and it does the infection, but because it's a pseudotype, it needs help. Then it's like, oh, how convenient, there's this protein for me here. And then it can now go off and do another infection.
What's an example of transfection, like electroporation?
You might. I have to double check if you can electroporate. I think you can. I think that would be classified as an example. A good example would be using lipofectamine. And Juan Carlos is mentioning Sendai virus. So yes, that's another example. So depending on how you do the transfection, you could call it 2 rounds of infection, or you could do one that is not considered to be infection. So if you look up lipofectamine 3000, that's a transfection reagent. That is not an infection and allows you to deliver genetic material to host cells. And so this is all very important because this is how they did most of their experiments, right? So they were able to use a system where they had a VSV virus that was missing the glycoprotein, and then they were able to help it out by providing glycoproteins of interest. So if you look at the screen, you see that there's VSVG and there's SARS-S, right? So they can just add whatever glycoprotein of interest they want, okay, by giving the host cell it first. So,
okay, so I have a question. It's not the genetic material that's in the host cell, right? The host cell has the protein, that's
host,
gonna attach to the virus instead of the genetic material that's gonna be able to basically form with the virus genetic material.
No. It doesn't. It's not the genetic material. The pseudotypes are still going to lack the ability to produce the glycoprotein.
So it's basically like a protein, either like SARS-S protein or the VSVG protein that is going to be like attached to the like empty capsule that the virus might have formed, and go ahead and infect other proteins.
I don't know what you mean by infect other proteins.
Like go ahead and which makes it have the ability to like infect other protein. I don't like other like cells.
Yeah, it would then be able to infect cells again. Yes.
Oh yeah, that's what I meant. Sorry.
Yes.
Thank you.
Yes, and Saad, you bring up a good point. Yes. Sorry if you missed my apology, but yes, it's all recorded. So I will probably modify this and just not provide any new content on Tuesday's class. But really, yeah, I mean, I could have definitely just ended at 4:15, but I'm doing this because I care about your education. So it's really I just care that, and I feel bad that you guys have not had class at a normal thing. So that's all there is to it. But I do apologize for not keeping on time. So anyway, that's the basic concept, and this is all recorded. I'm going to take a quick break to transition to office hours. I just need to get some water because I've been talking for a while. I think that is all. Oh, the last thing I want to show you is this is the genetic model for the original vesicular stomatitis virus, and then this is the modified version that they generated. As you can see, they replaced the glycoprotein with GFP and luciferase. This is the firefly luciferase. And then they have additional proteins as well. Let's see. Okay, so that's, and I can send a reference for this. It's actually, yeah, there's a couple interesting papers that go into more depth about it. All right, so for the office hours, I'm just gonna stay here. I don't even wanna think about creating new things. So if you want to leave, totally won't be offended in the least. If you want to join in, great. And if you aren't sure, then flip a coin. I don't know. All right. Everyone stay safe. Thanks for coming. And please respond to the surveys I create on if you like this better, if you like this worse. And we will go from there. All right. So yes, be safe. I'll be back in 5 and we'll do some office hours. Okay. Howdy, folks. Let's see, have some more chat stuff.
Dr. Adams, can I ask a quick question?
Yes, you can.
So I've been trying to access your zip folder on Canvas for yesterday's or the day before's lecture.
The zip folder?
I don't know what it was called. It said something. Let me look on it.
Like the zip drive, or okay, yeah, like the zip. Yeah, and okay, the file with the recording.
Under modules for 317, it's the let's see. It's the instructor slides 3-1720 zip.
Yeah.
And when I download that, I get a file downloaded to my computer, and it has the instructor slides, and then it has 3 recordings. It looks like.
Yeah. Sorry.
They're a .trek, and for my Mac, I don't have a application that I can open, so I haven't been able to access the lecture for that.
Did you try going to the link I posted via Google Drive?
Google Drive.
Or the YouTube link that I sent out this morning.
So yeah, and then that was another thing. Someone mentioned something about YouTube. I was talking to some people in classes today, and there have just been so many updates, which I totally understand, but there have been so many updates with different information. I'm just having trouble kind of keeping up with the assignments and what's going on.
Right.
So you posted this to YouTube, you said?
Yeah. So yeah, I really apologize for all the updates. I know it's a lot, and I wish there was like a way that I could condense these things, and I think it'll get better, with each class, and once we start to get some stability here with the coronavirus. But I think that the key thing is that if you look at today's updates, we have a link to the YouTube video, and I think I'm just going to try to always just do YouTube in the future, unless Zoom is really easy, so you don't have to worry about the zip files. Okay.
Yeah, great. That's awesome. And then, so can we subscribe to your YouTube channel and then just expect the lectures to be posted there all the time in the same spot?
Yeah. So that's my goal, is to just do the YouTube thing. So you will just have all that information, and you don't have to search through 20 updates. And so
yeah, that's awesome.
Do you know if you're able to subscribe to the YouTube channel right now?
Yeah, so I'm right here right now. I just hit subscribe and it says I'm subscribed.
Okay. Great. So sometime tonight or tomorrow, you should just see this video posted there. And if you don't, feel free to send me an email, and then I can troubleshoot.
Okay, cool. And then just to recap on our assignments starting from here on out, we have the pre-lecture photo 19, and then the post-lecture assignment, which is it's going to be like a set of questions or something like that we
right.
And then is there another discussion, or are we just doing that post-lecture assignment, posting that to a discussion board?
Yeah. So I've transitioned. I've been trying to transition to discussion boards because I feel like they're more communal, and instead of just everyone submitting assignments in secret to me, you can once you post, you get to see whatever other people did and sort of learn from your peers. So that's why I've done this sort of discussion board emphasis. But basically, my goal for this structure is to have a post-lecture assignment for every class, and also the photo sharing thing. So I haven't finished this lecture's post-assessment, and I'm probably going to modify it since I know, we went over time and stuff. But basically, that's just sort of the goal, to have the assessment of the lecture, and that's really to encourage people to not just blow off lecture and then marathon watch 10 lectures in a row or something.
Right.
I wouldn't wish that on anyone, but that's sort of the idea.
Cool.
And so that's the rationale. And then I'm gonna take some time over spring break to think about if we're gonna continue with the micro reviews for like the reading. I see Kara's asking about the pre-lecture assignment. So far, there haven't been any of those since we went online. But I just didn't want to like have too many moving parts, and I felt like having a pre-lecture, a post-lecture, and a discussion board with photos is like it's just a lot to keep track of. So I don't want to make your lives overwhelming either.
Cool. Yeah, that's awesome. So one last question here. So a lot of my classes are kind of in the same boat, and there's been like delay in posting and stuff like that.
Yeah.
And I'm writing out all my new assignments and stuff like that, and their due dates, and they're kind of all starting to pile up towards like this weekend. Are you going to be flexible on due dates at all? I'm trying to prioritize work for different classes, and then also deal with stuff here at home.
Right. Yeah. So they told me that I should not be flexible at all, and I should make everything due right now. So if you could just submit it, that'd be great. Yeah. Of course. So like, I think I've set like 1 or 2 due dates, but like honestly, everything is very flexible. Like I've also had to deal with family things, and like my wife has had to deal with family things. Like we all are like doing the best we can. So honestly, like there's like the most flexible, like I don't want to say like it's impossible to turn something late in, but like please like try to just like if something comes up or like you have like all this other stuff that's piling, like it's totally fine. All I'd ask is that we can try to get the assignments in within like 2 or 3 days of when the like target due date is, and if you need more time, like just let me know. But it's really just it becomes like an organizational thing for me, like just to make sure I'm like, okay, like can I move forward to the next assignment as everyone's sort of completed things. So it's really just more organizational. Like these deadlines are all somewhat arbitrary, but I'm definitely going to be very lenient on that, and that goes for everyone.
Much appreciated on our part for sure. Cool, thank you. That was all the questions I had. Thank you so much.
Yeah, you're welcome.
Hello, Dr. Adams. I have a question too, actually.
Great. Yeah.
Yeah. So basically, how is it gonna be the format for like the Canvas exams? Like, do you like so you're gonna ask us like graphs questions and like make us like analyze that, or is like more like you gotta draw it out and then like on Canvas?
Yeah. So there's gonna be a combination of those, and then also like some things will just be like sort of like what describe this process, or like describe 3 out of 5 steps for DNA replication, or like what are the metabolites that are generated from beta-galactosidase. Like some of these questions will definitely be, I think, very easy, especially with open notes. I think some of them will be medium, and then some of them are gonna be hard. And the format is, I don't know if you saw, I know I've had a lot of updates, but like, it's not gonna be like, you're gonna submit it through Canvas, but like you can submit it as a Word file, as a PDF, as a PowerPoint. You can use a combination if you want, or you can even just take a picture of your exam.
Okay, so it's gonna be like written basically. Oh, okay, yeah, for sure. And one more question. So are you gonna have more office hours like throughout the week, or is this gonna be like after class? Like,
so I think I might change my times now that we're all remote, because I had one student on Wednesdays from 8 to 9, and that used to be very popular. So I'll probably make another poll for like, gosh, there's gonna be like 20 polls. Polls for like poll for office hours. But yeah, like I mean, they're for you, and I figured I might be able to catch a decent number of you after this class. But I also have no problem moving things around to like a morning time or a midday time. Like my schedule now has a lot more flexibility than it would normally have. So I'll
So
find
we
a
can
suitable.
Okay.
I'm sorry.
We can just email. We can just email you, right? About like.
Yeah, you can also email me for sure. Yeah.
Okay, for sure. Okay. And yeah. So for the discussion tomorrow, do you just want us to like? Is it just basically like QA answers, or just like you're gonna give us like list of questions and then just answer from there?
Yeah. So if you had a chance to look at the post-lecture assignment for Tuesday's class, like it's 4 questions as a PDF, and like you can take a picture, you can write it in Canvas, you can write it in Word, like however you want to do it is fine. Like I'm very flexible with the format, especially because some of these are graphing. So like I want you to, if you're super PowerPoint savvy and you love to make those graphs, great. If you're you like to hand draw things, great. Like whatever works.
Okay, sounds good. Thank you.
Other questions? Oh, okay, chat. I want to ask questions about my last whether this exam I was going to come in person and then school at down. Would it be best to ask on here or separately? Okay, hang on. Kara, you can ask them here, or if you want to set up a separate time, we can do that separately. I still am happy to make separate appointments for people, so we just need to set up a time that works. And yes, Sherry also pointed out, if you like, the one thing I know how to do is direct message people, which is like you hit the everyone tab and then you scroll down to my name and then you can direct message me. Other questions? Sorry if I missed some in the chat. Okay. Let's see. Other questions? It looks like there's one on viruses. Yeah, unfortunately, going over pseudotyping did not go nearly as well as I wanted it to. And Evelyn is asking graphing. Yes, there definitely will be graphing, and then there will be explaining in short answer. And then there will also be like some questions that are just one word. One new message, okay. Okay, great. Let's see. Yeah, so the virus section did not go super well today, but I will try to figure out like maybe an easier medium level question from the pseudotyping stuff, and then put that together for the micro exam that we talked about, and then we can practice that together as a class. Other questions either via audio or chat? And if you don't have any, it's totally fine, but I'm gonna be hanging out here for a bit. For polling option, you gotta go to Zoom. Oh, thank you, Sherry. Awesome. Oh yeah, I was gonna play a song today, but ran out of time. It was gonna be a little help from my friends, or our friends, that Beatles song, I feel like we're all helping each other through this crisis, so I thought it was appropriate. Writing that down. Zoom.us/profile/setting. Okay, I'll look at that. 2 new messages. Turn on polling, boom, great. Oh, that's gonna be awesome. Thank you, Sherry. Technology.
Wait, Dr. Adams.
Hello.
Yeah, so you want us to draw like the virus after it like incorporates the GFP or luciferase gene for the activity?
I mean, I was trying to do something creative and fun. Like, I'm not going to ask you to draw the virus for the test. But like what you could do is, I was trying to have a group like art collaboration where you could draw the virus, and then see what was missing and how that might impact things. But at this point, it was also important to understand what was missing from the virus and also like what was added to the host cell for the system to work. So yeah, unfortunately not go quite as planned, but if you want to try and draw the virus, you can.
Okay. Because I saw like I got a Canvas notification that says like submit like a screenshot of it.
Yeah, that was the plan, but I'm probably it's just an optional thing. If you want to draw it, you can. If you don't have to. Like if we had gotten through this faster, then it would have had more time and we could have actually shared artwork and stuff. But yeah, I'm learning that online lecture is sometimes a little slower than the in-person one.
Oh, okay, thank you.
You're welcome. Is that the post-lecture assignment? Sherry, is the polling the post-lecture assignment, or is there's definitely going to be like a question on like the paper that we went over. It's not just going to be polling. So it'll be a combination of things.
In my other class, there were some problems with the graphing during the exam. So I was wondering, would taking pictures of the graphs that we draw be okay and emailing them to you? Or
Yeah, you can take pictures of the graphs. You can take pictures of what you write. I'm not asking anyone to generate graphs via Canvas. I'm asking people to generate graphs via PowerPoint or by drawing them, or if you have Excel or another graphing software program, those are all acceptable options.
okay, thank you.
You're welcome. Other questions, clarifications, compliments?
Oh yeah, Dr. Adams.
Yeah.
I also have a question, not related to class, but with this like coronavirus issue, are we still gonna do the summer research thing?
In my lab?
Yeah.
Yes, I just don't know when they're gonna let students come back on campus.
Okay.
So basically we're all sort of in a holding pattern on research, but I'll probably move forward with setting up interviews towards the end of the semester, and then for those students that are still interested and then seem like a good fit for the lab, we'll definitely have people in the lab during the summer. So just health and safety is number one, and then if we can move forward with the research, then that's awesome.
All right. Thank you.
You're welcome. It's pretty cool.
All right. I think that's about it. I'll see you later.
All right, take care.
Have a good day.
You too.